Remember that the first phase is glycolysis and it takes place in the cytoplasm, but the second and the third phases that we will be talking about today take place in the mitochondria. And before we get into the processes themselves, I want to orient you to where we are. So this is a mitochondria. There are two layers of membranes. There's the outer phospholipid bilayer and an inner membrane, which is also made of phospholipids. And an interesting aside is that scientists have a lot of evidence to support the idea that that mitochondria were once free-living bacteria, and the outer membrane is the membrane of the cell that engulfed them that they are now living inside of. It's called the endosymbiotic theory, and in fact bacteria also have their own DNA and can replicate on their own. The liquid filling on the inside of the mitochondria is called the mitochondrial matrix, and it is where the Krebs cycle occurs. And the inner membrane is highly folded, and the folds are called the cristae, and because it is so folded, it provides a large surface area for the third part of aerobic respiration, the electron transport system, to occur across that membrane. So phase 2, and remember we're in the mitochondrial matrix, has several names. The most popular name is the Krebs cycle. It can also be called the citric acid cycle because citric acid is the first large intermediate in the cycle and also the last, so it starts and ends with citric acid. And it is sometimes also called the tricarboxylic acid cycle because citric acid has 3 carboxyl groups. There is one carboxyl group, there is a second carboxyl group, and there is the third carboxyl group. So this first set of reactions up at the top of this diagram show you what most textbooks will call the transition reactions. And when we are going to be summarizing all of the totals of the molecules produced during the Krebs cycle, we're going to include the numbers from the transition reactions. So remember that we have pyruvate molecules, which are 3 carbon chains, which are the products of glycolysis that are going to be entering the mitochondria. That actually requires the cell to spend some ATP, so the number of ATP that we've previously told you, that 38 molecules of ATP, is actually unrealistic because the cell has to spend some ATP to move pyruvate and actually also ADP into the mitochondria. So we start off with pyruvate, and it is converted into a 2-carbon molecule called acetyl-CoA. It's an acetyl group with a coenzyme A attached to it. And in between that step, we lose a carbon and hydrogen. And in fact, actually, carboxyl groups are removed, which is why every time in this cycle, you'll notice everywhere where a carbon dioxide is produced, NADH is also produced. NADH stands for nicotinamide adenine dinucleotide. And I have a handout for you that will explain exactly what NADH is, and it shows you the structure. So now we have 2 molecules of acetyl-CoA, and each molecule of acetyl-CoA enters the citric acid cycle. So before we enter the citric acid cycle, we have already produced 2 carbon dioxide molecules and 2 NADH molecules. Now each of the acetyl-CoA molecules enters the citric acid cycle. So when we are coming up with the totals of all of the molecules produced, we have to count everything in the transition reactions once, but everything in here twice, because all of these numbers, if we're counting, are for each individual acetyl-CoA molecule, and we have 2. So the first thing that happens is that an enzyme joins a 4-carbon molecule of oxaloacetate to acetyl-CoA to make citrate. By the way, as I have previously explained to you, anything that ends in ate can also be ic acid. So another name for oxaloacetate is oxaloacetic acid. And another name for citrate is citric acid. So we have a 4-carbon molecule plus a 2-carbon molecule make a 6-carbon molecule. That 6-carbon molecule is converted into an isomer of itself by an enzyme, and that molecule is called isocitric acid or isocitrate. Now, isocitrate is converted into alpha-ketogluteric acid or alpha-ketoglutarate, and we go from a 6-carbon molecule to a 5-carbon molecule. Anytime we are losing carbons in the carbon chain, a carbon dioxide is being produced and NADH is also being produced. The interesting thing about alpha-ketoglutarate acid is that if we actually were metabolizing fats or proteins, they actually enter at this point of the Krebs cycle, and of course would skip glycolysis because they're not glucose. At this point, we now have a 5-carbon molecule that's converted into a 4-carbon molecule, and that 4-carbon molecule is called succinyl-CoA, and of course carbon dioxide and NADH are also produced. It is at this stage between succinyl-CoA and succinate down here where our ATP is produced. And actually first, before adenosine triphosphate is produced, guanosine triphosphate is actually produced, which is converted into ATP. That's not something that we test you on. I just wanted to explain why you see GTP instead of ATP right away. The rest of this is mostly for regenerating oxaloacetic acid, although we do get some additional NADH produced. So in this stage, from succinate to fumarate, or succinic acid to fumaric acid, a new molecule that we haven't been talking about yet called FADH2 is produced. It is a good energy carrier, but it is not quite as high in energy as NADH, and in this stage there's not enough energy to produce NADH, and we get FADH instead, and I will be showing you very shortly what FADH is used for. And then fumarate is converted into malate. Malate is converted back into oxaloacetate, and at this last step we get some additional NADH. So when we count up all of the products of the Krebs cycle, the first thing we want to account for is how many ATP. Now we got 1 ATP per acetyl-CoA, but we actually had 2 acetyl-CoA molecules, so the number of ATP produced would be 2. If we're going to total our NADH, we have to count the 2 from the transition reaction, so 1, 2, and then 3, 4, 5, 6, 7, 8, because remember that we have 2 times everything in this cycle. So we get 8 NADH produced. Now when we're talking about how many FADH2 we get, we see it showing up once in the cycle, so that means we get 1 for each acetyl-CoA, but we have 2 total. And then the total number of carbon dioxide released, if we count the transition reactions, we have 1, 2, 3, 4, 5, 6, and that's it. And if you remember that all of the carbon dioxide that's made during aerobic respiration is produced during the Krebs cycle, it will be easy for you to remember how many CO2 are produced, because there are 6 carbon in a glucose and 6 1-carbon molecules are produced when it's completely broken down. Obviously we know the ATP can be used by the cell to do work, but we're going to see what happens to the NADH and the FADH2. They can be used in the electron transport system to make even more energy available for the cell. Now I'm going to discuss the third and final phase of aerobic respiration, and it is called the electron transport system, or ETS for short, and sometimes it's also referred to as the electron transport chain, or ETC. Now we've zoomed in on the inner membrane on the cristae, and in this area of the diagram we would have the matrix, and this area would be the area or the space between the 2 membranes. The outer membrane would be up here. Of course, we're not showing that in this diagram. So these blue objects in this slide are actually enzymes. The ones toward the end are referred to as cytochromes. The beginning ones have specific names there, which you would have to learn if you were in an upper level biology class or if you take AP biology before you graduate, but we're not going to worry about the names of those enzymes right now. This process is going to look very familiar to you if you remember the light reactions of photosynthesis. So in photosynthesis, we had energy from the sunlight energizing electrons. And in this process, we have the energy in these energy molecules, NADH and FADH2. They are donating electrons, and the energy from the electrons is going to be used to pump hydrogen ions against a concentration gradient from low to high concentration into the space between the 2 membranes. That would be considered active transport. NADH can power 3 proton pumps as the electrons are passed along. And at the final cytochrome, it's often referred to as the terminal cytochrome, the electrons are rejoined with hydrogen and oxygen to make water. And this is a non-toxic waste product of aerobic respiration. And if you notice, the oxygen that's used in aerobic respiration doesn't come into play until the very last step of the very last process. Oxygen is often referred to as the final electron acceptor and sometimes referred to as the final hydrogen acceptor, although probably less often. FADH does not have as much energy as NADH, and FADH2 actually skips the first enzyme and starts at a later point in the electron transport chain, and so therefore can only power 2 proton pumps. And once you have this hydrogen ion concentration, you actually have an electrochemical gradient where you have a more positive charge on this side of the membrane and a more relatively negative charge comparatively on this side. And also the molecules are going to tend to want to diffuse from high to low concentration in any case, even if they weren't ions. They're going to diffuse through an old friend of ours, ATP synthase. And remember that just like in the light reactions, that flow of hydrogen ions through ATP synthase actually spins a mechanism inside the ATP synthase, forcing adenosine diphosphate, also known as ADP, together with a phosphate to make ATP. And so for every NADH, 3 pumps are powered, and therefore we get 3 ATP for every NADH. Because FADH only powers 2 pumps, we end up getting 2 ATP molecules for every FADH2. This diagram I like because it shows where the hydrogen ions are being actively transported into the space between the 2 membranes, and then the facilitated diffusion happens, so the hydrogen ions flow back into the matrix. The one thing I would caution you is that, of course, this orange inner membrane would be folded, but it was very hard in the diagram to get that to happen. So just beware that this orange molecule, this orange phospholipid would be a phospholipid bilayer and would be highly folded. So just once more again, the NADH powers 3 pumps, and therefore 3 pairs of hydrogen ions are pumped into the space between the membrane. And FADH2, which is shown in red on here, powers 2 pumps. And in either case, the electrons and hydrogen ions are joined with oxygen to make water at the end. And without that, you'd have a buildup of electrons and hydrogen ions, and the whole process would really stop. So electrons gathered in the Krebs cycle and glycolysis in NADH, and in the Krebs cycle and FADH2 are used to drive the movement or active transport of hydrogen ions. And for each NADH entering the ETS, 3 pairs of hydrogen ions are actively transported, and for every FADH2, only 2 pairs of hydrogen ions are actively transported. And then at the final terminal cytochrome, the electrons finally react with hydrogen ions and oxygen to form water on that inner membrane surface. So if we are totaling the total number of ATP produced during aerobic respiration, if we include the NADH from glycolysis, which is 2, and we had 8 NADH from the transition reactions and the Krebs cycle, which was 8, and we had 2 FADH from the Krebs cycle. Now remember, for every FADH2, we get 2 ATP, so 2 times 2 is 4. And then if we add the total number of NADHs that are produced during glycolysis and Krebs cycle, we get 10. 10 times 3, because remember, NADH powers 3 pumps, we get 30 ATP. And if we add 30 and 4 together, we get 34 from the electron transport system. Now, if we're going to add that to the ATP previously produced, we would get 2 ATP from glycolysis, 2 from Krebs cycle, and 34 from the ETS. And if we total this all together, it's a total of 38. Of course, this number is really an overestimate, because if we go back to the previous slides here, remember that we have pyruvate and it's not in the mitochondria, and pyruvate and ADP actually have to be actively transported into the mitochondria. So some more ATP would actually have to be spent. Scientists estimate that with all of the active transport that has to go on, that overall it ends up being closer to 30 ATP molecules, but we're going to go with the number 38 when we test and quiz on this.